Now we're going to look at lipid synthesis, or the anabolic pathway of the metabolism of lipids. Here are some questions that you should be able to answer when we're done with this unit. How are fatty acids synthesized? How is cholesterol synthesized? How are lipids transported throughout the body? And how are steroid hormones synthesized and utilized? Before we take a look at synthesis, I want to compare two molecules. One you're quite familiar with already, coenzyme A. But in lipid synthesis, we're going to see this molecule, ACP-SH, acyl carrier protein. In comparison of these two molecules, I want you to look at them from this end, or this segment. They are exactly alike. This and this are the business ends of these molecules. In coenzyme A, we have an ADP residue, whereas in our acyl carrier protein, SH, we have a carrier protein attached to this point. This protein is attached through a serine residue. And I guess that's about it for this slide, and moving on. So this is a big picture, and this is called the citrate malate pyruvate shuttle. It provides acetyl-CoA and NADPH for fatty acid synthesis. So this is just another slide or figure that shows how these processes are linked to one another, because here we're seeing the citric acid cycle, and over on this side of the slide we're going to have the activation of fatty acid catabolism and also fatty acid synthesis. So these processes both begin in the cytosol. We've already covered catabolism of fatty acids, where those fatty acids are activated by being linked to the CoA enzyme. They are then converted to that carnitine derivative, which allows passage through that intermitochondrial membrane. After passage, if you recall, this is reattached to a CoA molecule and then can undergo fatty acid oxidation to produce acetyl-CoA. So, how are these linked? Acetyl-CoA, can't cross the membrane. So it has to be converted to citrate. And this is to regenerate fatty acids, or synthesize fatty acids. So citrate passes through that intermitochondrial membrane and can be converted to acetyl-CoA and oxaloacetate. Acetyl-CoA then can be utilized, or the 2-carbon utilized, can be utilized to produce fatty acids. You'll notice there are blue arrows and red arrows on this figure. The red pathways provide electrons for fatty acid synthesis. So here we see the generation of NADPH. Remember, this is often involved in synthesis reactions, so that is the fuel with respect to electrons. The blue pathways are providing the carbon units for fatty acid synthesis. So the formation of malonyl-CoA is going to commit the acetate units to fatty acid synthesis. Acetyl-CoA carboxylase is the enzyme that catalyzes the carboxylation of acetyl-CoA. For this process to occur, we need some bicarbonate, we need some ATP, and we need also biotin. So this is a biotin-dependent reaction. And what happens is we get an intermediate, this carboxybiotin enzyme. So we have actually attached, this is our CoA, and we have carboxylated that molecule with the CO2 from our bicarbonate. So here is the carboxylation. The end of this reaction then produces malonyl CoA. So there is a separation or cleavage from this, this acetyl-CoA carboxylase that gives us our malonyl CoA. Now we're going to take a minute to look at the regulation of acetyl-CoA carboxylase, and it is regulated by phosphorylation and dephosphorylation. So under conditions of abundant metabolic fuel or high energy, the products of carbohydrate and amino acid catabolism are directed toward fatty acid synthesis, and that results in fatty acids being stored as triacylglycerols in those adipocytes. The unphosphorylated acetyl-CoA carboxylase binds to citrate with high affinity, and thus it is active at very low concentrations of citrate. Conversely, high levels of fatty acetyl-CoA is going to inhibit fatty acid synthesis. That means we already have a lot in our system. So also involved in this process, obviously, is ATP, which is going to provide that phosphate group, and kinases that will transfer that group to our enzyme. And to reverse this process, we need a phosphatase. Phosphatases, if you remember, dephosphorylate. Phosphorylation of the regulatory sites, as we see down here, decreases the affinity of the enzyme for citrate. So it requires very high concentrations of citrate to overcome the inhibition and reactivate this enzyme. So the first step in fatty acid synthesis is the production of malonyl CoA. The second step requires two parallel reactions. In the first, we have acetyl-CoA, which, whoops, I've lost my little pointer here. Here we go. Acetyl-CoA, which is going to be transferred, or the acetate group or acetyl group is going to be transferred to an acyl carrier protein. And this is accomplished by the use of this enzyme, acetyl transacyclase. So we get an acetyl ACP unit. The second reaction that occurs is switching out that CoA to the acyl carrier protein. So both acetyl-CoA and malonyl-CoA are then converted to ACP derivatives. And these products are going to condense and react with one another, or I should say react with one another in the next step of this process. So here we see the complete cycle of fatty acid biosynthesis, in which there are 4 steps. This is a cycle that you are going to be responsible for. I'm not going to hold you responsible for cholesterol synthesis, but this one is a little simpler. So we're starting with our malonyl ACP unit and our acyl ACP. So in the first step, we are going to be condensing these 2 molecules, and that's accomplished by the acyl-malonyl ACP condensing enzyme. That's a mouthful. We get the release of one of those ACP carrier proteins, and we see also a decarboxylation. The condensation, or the energy that is required to accomplish this condensation, is provided by the release of that CO2 molecule. Another aspect of this reaction that provides energy to create favorable thermodynamics is the release of the ACP. So cleavage of that thioester bond also has a high energy of hydrolysis. What we produce then is this molecule, and this is a beta ketoacyl ACP protein. The ketone group in the second reaction is going to be reduced to an alcohol with NADPH serving as the reductant. So we need NADPH, we also need a hydrogen ion, and the enzyme beta ketoacyl ACP reductase. So we are reducing this, this carbonyl group to an alcohol group in this step. In the third reaction, we are going to release a molecule of water and introduce a double bond. So this is a dehydration reaction, and we are going to form a trans double bond here at this point in our molecule. This requires the enzyme 3-hydroxyacyl ACP dehydratase. And in the fourth reaction, we are going to reduce that double bond here to a single bond. And in effect, what we produce is a 4-carbon butyryl ACP. So we have added 2 carbons to produce a 4-carbon acyl group. This acyl group will then be condensed with another malonyl ACP in the second round of biosynthesis. So each successive round elongates the acyl ACP unit by 2 carbon atoms. Because fatty acid synthesis occurs 2 carbon atoms at a time, most of the naturally occurring fatty acids contain an even number of carbons. So what we're looking at here is the number of cycles and reactions required to produce palmitate or palmitic acid. And if you recall, that is a 16-carbon saturated fatty acid. So in that first reaction, we produce that 4-carbon ACP, acyl, and after 7 reactions, we will be able to produce, or we will end up with palmitate or palmitic acid. Now, this can remain as palmitic acid if we need that, but we can also use this to generate longer fatty acid chains that can be saturated or unsaturated, and this occurs through the actions of enzymes such as elongases and desaturases to introduce those double bonds. This figure just highlights the reactions and the players in beta oxidation and biosynthesis of fatty acids, keeping in mind that beta oxidation occurs in the mitochondrial matrix, while biosynthesis occurs in the cytoplasm. At the end of this unit, I will show you another slide that compares and contrasts and highlights the players that make these processes thermodynamically favorable. So that's all I'm really going to say about this. This is for your benefit in taking a look at the differences and similarities with these two processes. Sorry about that. We appear to have a duplicated slide here, so we're going to move on. I'll have to remove that later. Now, we've seen that acetyl-CoA carboxylase has been or is regulated by phosphorylation and dephosphorylation. But what triggers that? And here's what we're looking at now, or that's what we're looking at now, is the hormonal regulation that's involved with activating or deactivating this enzyme. So we're looking at the hormones insulin and glucagon, keeping in mind that insulin is secreted and signals pathways when energy is high, and glucagon is that hormone that maintains glucose homeostasis or energy when we are in between meals. So I don't think you've seen the degree of this degree of an example in the membrane, and you're not going to be responsible for this, but there are receptors in the plasma membranes of cells. Glucagon binds its receptor just like insulin would bind it. When glucagon binds, we have the activation of cyclic AMP, that second messenger, which is capable of acting this protein kinase. Once that kinase is active, we can either turn on or turn off lipid synthesis or lipid degradation. So in times of high energy, we do not need to generate or degrade fatty acids and glycerol. In terms of high energy, we're going to synthesize fatty acids for storage. On the other hand, glucagon is going to activate the pathway that releases and degrades those triacylglycerols. So what we're looking at here is the molecule cholesterol. So we're all familiar with cholesterol. It has some positive roles. It's a membrane component, lots of cholesterol in plasma membranes or cell membranes. It's a precursor for steroid hormones and bile acids. But on the other hand, it also can, if we have too much cholesterol, be a negative, have a negative capacity. And that is because it's a major component of the fatty plaques that are found in atherosclerosis. I'm not going to go into the detail of these carbons, these red carbons here, but essentially what this is trying to show us is that the backbones or the backbone of this cholesterol comes from, carbon atoms from acetyl-CoA. So it's basically formed by the polymerization of acetyl-CoA molecules. We are, however, going to look at cholesterol synthesis, and again, you're not going to be responsible for these reactions. And when it's all said and done, I think there are about 28 reactions required to produce cholesterol. But we can break cholesterol synthesis into 4 stages. And this slide does not show you the multiple steps in each stage that are catalyzed by different enzymes. But stage 1 begins with a 2-carbon compound, acetyl-CoA, and ends with a 6-carbon intermediate, mevalonate. So here's the product of that first stage. Stage 2 begins with that 6-carbon mevalonate and ends with a 5-carbon activated isoprene analog. In this third step, 6 of those activated isoprenes are polymerized to give a 30-carbon intermediate, which is squalene, and then in stage 4, squalene is converted to cholesterol by a series of rearrangements and ring closures. All right, so this is the process or the reactions that are required to produce mevalonate. In the first reaction, we have 2 molecules of acetyl-CoA condensing to form acetoacetyl-CoA. In the second reaction, we're going to introduce another molecule of acetyl-CoA to produce 3-hydroxymethyl-glutaryl-CoA. Finally, we're going to have the thioester carbonyl reduced to an alcohol, and for this we need 2 NADPH because this is a 4-electron reduction, and we're also going to see the release of the CoA molecule. The driving force of the reaction, this reaction series, is going to be the release of the coenzyme A molecules and the conversion of 2 NADPH to NADP. And again, our product is mevalonate. So in that second phase, or maybe not second phase, but next set of reactions, and again, I'm not going to go into the detail here. I just want you to see how complicated the synthesis of cholesterol is. We first are going to convert mevalonate to a pyrophosphate derivative, and this is a 10-carbon pyrophosphate. This requires 2 ATP, and again, these enzymes are kinases, so they are phosphorylating these molecules. The second reaction involves also an AT-dependent dehydration and decarboxylation. So we are dehydrating and decarboxylating. So we've lost this carboxyl group. They're not showing the loss of the water molecule, but we get the formation of that double bond where that occurred. Then we undergo an isomerization, and here we have an isomerase that does this. And finally, we have 2 molecules of this product or intermediate that condense to form the geranyl pyrophosphate. And we also release inorganic phosphate at that time, so that is providing the energy to drive this process. So the series of these reactions are energized by the cleavage of those 3 ATPs, a decarboxylation, and the release of that inorganic phosphate. In the next step of cholesterol synthesis, we are going to condense a 5-carbon molecule with the product from the previous slide, which is a 10-carbon molecule. This involves a transferase. It also involves the release or the cleavage of 2 inorganic phosphates. So in this condensation, we are going to end up with a 15-carbon molecule. The cleavage of those phosphates is also what promotes that forward reaction. And in the final steps of cholesterol synthesis, we are going to condense 2 of the 15-carbon molecules. We also have the involvement of NADPH and hydrogen, and we produce the squalene molecule, which is a 30-carbon molecule. We're going to do a little more rearranging. We're going to introduce some double bonds. We're going to form rings. And our final product is that cholesterol molecule. So somewhere along the line in this process, we have 30 carbons here. We're going to lose 3 carbons as CO2 molecules. A double bond is introduced into one of the 6-membered rings, and one of the rings is hydroxylated. And so that leaves us with the final 27 carbons of cholesterol. Cholesterol has 3 fused 6-member rings, 1 fused 5-member ring, and an 8-carbon hydrocarbon side chain. And lastly, the slide I mentioned earlier, the comparison of the differences between fatty acid synthesis and beta oxidation. I think this is important for you to know, so I suggest you study this. And there are basically three of these differences contribute to the favorable thermodynamics of both pathways. The differences that change the energetics are: one, fatty acid synthesis involves a biotin-dependent decarboxylation reaction. Two, a malonic acid derivative is a participant in synthesis but not oxidation. And three, all of the redox reactions in synthesis use NADPH from the pentose phosphate pathway, and beta oxidation uses NAD plus and FAD. So synthesis is a reductive process versus oxidation, obviously, is an oxidative process. Synthesis occurs in the cytoplasm. Oxidation occurs in the mitochondrial matrix. We have our acyl carrier protein versus coenzyme A. We have as our, you could say, activating molecule, malonyl ACP versus acetyl-CoA. And again, the differences with our electron carriers, NADPH versus NAD plus and FAD. And we have a 3D hydroxyacyl in synthesis and a 3L hydroxyacyl in oxidation. So this concludes our discussion of lipid metabolism. Sorry, we're going to move on to amino acid metabolism.